professor, physician, epidemiologist. I am Dr. Sri Banerjee. Hello, everyone. I'm Dr. Sri Banerjee, core faculty for the College of Health Sciences and Public Policy at Walden University. And in this video, I wanted to go over a lot of the progress that has been made in genome-wide association studies. Honestly, if you're a budding researcher, if you're a new researcher, you should be looking into molecular epidemiology very early on. This is something that is, the tools are available. There's a lot of information and data that's just floating around out there, waiting to be analyzed to find out more information about. Currently, there's variants that are being identified and everything, and so I'll go over to some of these tools over the course of actually multiple papers and articles that have been generated surrounding this topic. And over the years, really over even the last 5 years, there's been quite a few tools that have been generated surrounding conducting genome-wide association studies. I actually had a conversation about these types of studies with one of my colleagues, who's a research coordinator at Walden University, and it's really, I think it's tragic that more places don't have capabilities to assess GWAS and, more importantly, polygenic risk scores. I also had the rare opportunity to recently actually collaborate on an NIH topic, on a NIH grant-funded research topic related to the polygenic risk score of obesity and a lot of the metabolic syndrome related items. Anyway, with all of that said, if I didn't introduce myself, I'm Dr. Sri Banerjee, and so here is now going to be, a little bit of a description talking about what the aim is for GWAS. So that's good to know first. And it's to identify associations of genotypes, simply. So associations between the genotype that's within place, so the CGAT code that's in place, and the phenotypes by testing for the differences in the allele frequency of genetic variants between individuals who are ancestrally similar but differ phenotypically. So it can be considered like copy number variants or sequence variants. So what I mean by that is it could be variants within a sequence. So you might, I guess I'm gonna do a lot of illustrations here. You might have a sequence of CGAT, and for those of you that might be new to molecular biology, CGAT are the 4 base pairs that are the building blocks for every human living existence. So you might have this kind of within variants that are within this base pair. So instead of a G, you might have a point substitution here. And depending on what that point's, so instead of a C, you might have a C. A, T, G. And to the naked eye, this may seem like a very minute sort of change. But trust and believe, there's scenarios and instances where that one base pair at the right, or should I say wrong place, has caused disease. And that one base pair will cause an amino acid substitution. And of course, here you have to know the central dogma of how DNA works. That's something you want to know. DNA is transcribed to RNA, and then RNA translates through transfer RNA to protein. And there's a set of 20 amino acids, and there's codons and anticodons. Another difference between DNA and RNA is that DNA typically has thymine as a base pair, and that thymine is replaced by uracil when you are talking about transcription. So this is the central dogma of how, so this is transcription, and I'm going to designate it by trans, and this down here is translation. I'm going to reset Zoom. We don't need Zoom. So that covers everything, really. The other thing that I wanted to point out is that there could be random substitutions which could cause premature stop codons, and all sorts of mutations that can take place. So not all mutations are bad. I think that's the other message here. There's a lot of different variants that can take place, that can occur, and that do occur, which a combination of variants can cause disease and phenotypic manifestations, but not always does it do that. So these sorts of kind of gray areas are what the reason is for the genome-wide dissociation studies. And this right here, the reason I'm reading this, and as you know, I've written primers myself, co-written primers. So this is a primer to how to conduct a genome-wide dissociation study. So if you're interested, if you want to know what this is all about, take a look at this. Linkage disequilibrium is a concept that has been described many times. And the idea is that previous GWAS have shown that most traits are influenced by thousands of causal variants that individually confer very little risk, and are often associated with many other traits, and are correlated with causal and non-causal variants that are physically close as a result of linkage disequilibrium, making direct biological causal inferences complicated. So even though they might not be like they might not seem like they're related causally, there's all of these linkages, and that's the linkage disequilibrium that we face with oftentimes in GWAS. Now, genomic risk prediction using genome-wide polygenic risk scores are helpful. For instance, coronary artery disease, if you're looking at AFib, type 2 diabetes, inflammatory bowel disease, breast cancer. I mean, all of these monogenic, in all of these monogenic risk prediction strategies are rare. So polygenic risk scores are more helpful. So of course the first part of this is genotyping, and finding out what type of genomes are out there. There's input files for GWAS, including anonymized individual ID names